Okay, in today's lecture, we will discuss how biologists use different genetic tools to study gene functions. Now we learned that many genetic diseases have gene alterations. In order to understand how these alterations contributed to the onset of these diseases, first we must understand the function of the gene product, meaning the proteins. There are several ways we can do this. We can artificially make more of the protein, which we call it overexpression. We can artificially express less protein, we call it knockdown, okay? Or completely wipe out the expression of the protein, which we call it knockout, okay? Or we can also make the disease-related mutations and study how these mutations change the function of the proteins. The genetic modifications can be carried out at different levels. If the change of gene is done using animal models, we call it in vivo, which is in contrast of in vitro works when animal or human cells are used. Those are the culture dishes, different culture flasks and dishes shown here. The ex vivo shown here means primary cells are isolated from animals or human beings and used in the in vitro experiments. Scientists also perform cell-free experiments using recombinant proteins or synthesized DNA and RNAs. Okay, so these are also in vitro work. Basically, in different model systems, we can overexpress, knock down, knock out, or make mutations and study the protein of interest. Therefore, the recombinant DNA cloning technology is an essential tool. One of the most common used tool in genetic modifications is plasmid. If you have worked in the lab, research lab before, probably you already know this. Okay, plasmids can carry your gene of interest and work as a basis to deliver any gene changes to your model system. The model system we talk about can be cell, can be animals, right? Plasmids are small circular pieces of double-stranded DNA that are usually 2 to 15 kilobases. Okay, in research labs, scientists can easily modify the nucleotide sequences of the plasmids. The plasmids can be amplified in bacteria, and the plasmid replication is independent from the bacterial's chromosomal DNA. So the table listed here show the several essential elements that can be found on plasmids, which you may have already learned from a genetic class. I just need to mention two elements here. One is this ORI, the origin of replication, okay? So this is where DNA replication of the plasmid starts, because the plasmid need to amplify itself inside the bacteria, right? So this is where it starts its amplify or replicates the plasmid sequences, okay? Another one is called MCS, shown here. This is a multiple cloning site. Okay, this is a short segment of DNA that contains a group of restriction sites. So these sites can be recognized and cut by specific restriction enzymes, allowing for deletion or insertion of additional DNA fragments. So if you want to modify a plasmid, change the sequences, how do you do this? You can utilize the MCS sites to insert or do some other modifications, okay? This slide gives you a general idea of how gene cloning is done in the lab. Okay, for instance, if you need to make a recombinant insulin protein in vitro, you need to first choose a plasmid vector that is suitable for recombinant protein expression. All right. For instance, shown here, this is the plasmid vector you want to use, and on top of that, you want to insert it in your target gene. For instance, the insulin gene is what you are interested. You need to insert it here. Okay, this can be done through the multiple cloning site. The empty vector, it shows the backbone vector, same thing. Okay, it will be cut open. Okay, this is handled by the restriction enzyme. And then the two pieces of DNA are joined together. So this join here and this part join there by an enzyme called DNA ligase. Okay, after ligation, the recombinant DNA can be transformed into bacterial and then replicate inside the bacterial. Okay, the plasmid vector also contains an antibiotic resistant gene, which allows the plasmid grow in a media containing antibiotics, such as like ampicillin or kanamycin. So the bacterial cells without plasmids will be killed by the antibiotics. But the bacteria containing the plasmid, they will be able to grow well on this antibiotic containing plates. They're forming colonies shown as these white dots. Let's take a more detailed look into the restriction enzymes. Those are the scissors, right? So these are bacterial enzymes. Scientists noticed that some bacteria resist viral infection because they have enzymes to cut the viral DNA into pieces. This led to the identification of these enzymes. Restriction enzymes recognize specific DNA sequences and cut the double-stranded DNA like scissors. But most of them produce overhanging single-stranded ends. We call them as sticky ends. Okay, let's see next slides for how sticky ends look like. So this slide shows several restriction enzymes and their cutting sites. These restriction enzymes have different names showing here, okay? And out of these 5 different enzymes, you can see only this enzyme called HAE3, it generates a flat cut. So basically the arrow shows where it cuts, and then this is the end product for this cutting. All right, so we call this as blunt ends because this is a flat in between, right? And then the other 4 enzymes generate overhangs, and we call these as sticky ends, okay? Because the sticky ends have complementary sequences. They always have one line here. These 4 are empty complement sequences. So if you have another piece of DNA with the same sticky ends, similar to this, but they are complementary to the other piece. So they will be easily ligated together with the help of base pairing, right? So that's why I call it sticky, not a blunt, okay? This slide shows bacterial cells containing plasmid DNA molecules on culture plates. So this is a culture plate, and with some culture media, this is actually LB agar inside it. And on top of that, there's bacteria containing plasmid DNA inside it, okay? So all these bacterial cells growing in the forms of colonies. Each colony is descended from one single cell. Therefore, each colony is a clone. All the plasmid DNA from one single colony is the same. So every single one, although you look it's quite big, probably containing like millions or even trillions of bacterial cells, but every single one is the same. If you would like to generate more plasmid DNA from one specific colony, you could pick the clone and further amplify the bacteria, and then harvest the cloned plasmid DNA from the culture. That's how you actually amplify plasmid DNA. Now we shift the gear a bit and talk about how you can amplify a segment of DNA from any DNA samples, all right? I'm sure that all of you have heard of PCR, also called the polymerase chain reaction. This is a very smart technology and became a revolution in cloning. Most importantly, PCR can recover DNA sequences from a very small amount of DNA, such as DNA from a single cell. So PCR has many cycles, and each cycle has 3 steps. One's a denaturation or hybridization, or also called annealing, and then, of course, extension or DNA synthesis. Let's watch a video on PCR.
The polymerase chain reaction is a method for making many copies of a specific segment of DNA, starting with a very small amount. This technique can be used to identify specific microorganisms from a small amount of DNA and to identify persons involved in crimes from DNA on cigarettes or in a single hair follicle. The DNA to be amplified is mixed with deoxyribonucleotides, a thermal stable DNA polymerase called Taq polymerase, and DNA primers. The DNA primers hybridize to the ends of the gene to be amplified and provide a starting point for the Taq polymerase. The mixture is heated to break the hydrogen bonds in the DNA, forming single-stranded molecules. The mixture is then cooled sufficiently to allow the DNA primers to anneal to each end of the segment to be copied. Taq polymerase then synthesizes the complementary strand of DNA using the primer as the starting point. The temperature is raised again to separate the DNA strands and then lowered sufficiently to allow the primers to attach. Taq polymerase now synthesizes another set of new complementary strands. This process is repeated until enough DNA has been produced to be identified or used for further research. After 21 cycles, one molecule of DNA can be amplified to over 1 million copies. This amount of amplification can be achieved by running the reaction overnight in a thermal cycler, an instrument that automatically raises and lowers the temperature at appropriate time intervals.
As the video mentioned, PCR doubles the amount of DNA with each cycle. So 30 cycle is actually a very common number for PCR reaction. Repeated cycles can amplify the original DNA sequence by more than actually 1 trillion times. Therefore, PCR is a very efficient way to obtain a large amount of DNA from little amount of DNA samples or DNA templates. After the invention of PCR technology, scientists have renovated the PCR and made it more quantitative and more accurate. Currently, there are 3 major PCR methods using in research labs. There are traditional PCR, we just talked about. So this method is almost only used for amplification of DNA segments. So qPCR is a quantitative method which can be used for a comparison of DNA or RNA level among different biological samples. Recent few years, scientists invented digital PCR, which is not only quantitative, but also it can provide absolute quantification and becomes more sensitive or more reliable than qPCR. This is a kinetic curve of a PCR reaction. The x-axis shows the cycle number of PCR, and the y-axis shows the relative DNA copy numbers. You can see the more PCR cycle produced, the more DNA molecules, okay? So interestingly, the amount of a PCR product has an exponential phase, linear phase, and then finally reach the plateau. So why the PCR reaction cannot be amplified, amplify DNA infinitely? Now think about it. So yes, because in this reaction, after 30 to 35 cycles, the building blocks such as the NTP and primers, they are used up, okay? And the PCR enzymes might be exhausted and lost activities. Therefore, the amount of PCR product is not increasing anymore. So for a traditional PCR, we usually collect the PCR product at the end point when maximum amount of PCR product has been amplified. So that's in the plateau stage, right? However, this cannot detect the amount of DNA existing in the original samples because every single sample will reach the plateau at the end point. In order to compare the different amount of DNA in different samples, scientists invented the real-time PCR machine. So real-time PCR or quantitative PCR and or something called qPCR are all referred to the same method, all right? So qPCR machine can record the kinetic changes of all PCR reactions. For example, this slide shows amplification of 10 different samples, and the amount of the template DNA used in these reactions has been labeled on this right part with different colors. Okay, so if you collect all the samples after 40 cycles, you don't see any difference because all the reactions reach the plateau. However, if you compare the curves at their exponential phases, with this blue line showing here, you can easily compare their different quantities. For example, the purple line, this line that contains only 0.1 picogram of template will need 33 cycles to reach this blue line, right? And then for the red line that contains, this is a 1 microgram of DNA template. And then it only let them about 10 cycles amplification, they can reach this blue line, this much of DNA content. So indicating these are very different, okay? So this assay, qPCR, has been widely used in research labs to quantify the amount of genomic DNAs or mRNA levels using the RT qPCR assay, meaning reverse transcription followed by a quantitative PCR. In recent years, another type of PCR method was invented named the digital PCR. The machine is quite costly. The entry level of this machine is about $100,000. So UH does not have this machine yet. And then I know that Baylor College of Medicine has one, and the MD Anderson Cancer Center has a few. Okay, digital PCR is special because it can give absolute quantification of the source DNA. The basic idea is that during the sample preparation, the machine can divide one reaction into many, many small reactions. This is a target content. This is a total amount. You see, they are divided into different fractions. And then each reaction can allow contain either no DNA template at all, like these empty wells, or you have a one copy of your template, okay? Then the PCR amplification will occur within these small reactions. After reaction is done, the machine will count how many small reactions have a DNA template, which show these bright signals over here. While no temporary reaction are showing as a dark, no signal, okay? So by counting these bright signals, you can tell how many DNA molecules exist in your original samples just by counting those peaks. Therefore, this method gives you an absolute quantification and is highly sensitive. and it can be used to quantify the amount of DNA or RNA when the source of nucleic acid is very little, all right? We just talked about reverse transcription, right? So from RNA converted into DNA. I know many of you already knew the concept, but I would like to discuss this in more details for those students who are not familiar with. We learned that PCR can be used to amplify and quantify DNA. But the PCR tag enzyme can only amplify DNA, but not the RNA, right? So because this reason, in order to quantify RNA, scientists took advantage of reverse transcriptase, meaning the enzyme that can reverse the transcribed RNA to make a DNA, all right? So showing here, this is a genomic DNA. We learned that genomic DNA can make RNA. And then the reverse transcriptase convert this mRNA into the sequence this is a sequence we call the cDNA. It's also meaning complementary DNA. So the amount of cDNA reflects the level of mRNA. Scientists can use the cDNA as the template to quantify the level of mRNA by qPCR method. Okay, then let me ask you a question. What is the difference between genomic DNA and the cDNA on their sequences? So the answer is here. Because the cDNA is derived from mRNA, it contains only exons but no introns, all right? So now we learn that cDNA can be used to quantify the level of mRNA by RT-qPCR analysis. Similarly, when scientists perform RNA sequencing to quantify all the transcriptome inside cells, the RNA has to be converted into cDNA before the sequencing can be done. We will learn the next generation sequencing in future lectures. But here, cDNA can also be used to generate recombinant protein. For example, we talked about generate the insulin protein in bacterial culture. How can we obtain the insulin gene to construct the plasmid vector? Yes, we need to get a cDNA sequence of insulin. One expression vector on expression vectors, we only use cDNA, but not genomic DNA. The introns in genomic DNA can be very long and interfere with cloning and expression. So we only use the cDNA if we wanted to use it for cloning to make genetic products. Okay, now say we have PCR amplified the target gene, and I would like to check the amount and the purity of my PCR product. In this case, we need to run an agarose gel. Then we will use this technology called the gel electrophoresis. Probably all of you already have the experience. In this experiment, first you need to make the gel, right? So you will weigh the agarose, heat it up in buffer, and pour it into a gel box and let it to cool down. Then do you know why we use agarose to prepare the gel? This is because agarose will form the pore-containing structure, and DNA molecules can pass through these pores. The more agarose you are using, the smaller these pores will be, right? So when you put the gel in the buffer on the connected electrode, you would like to put the sample side close to the negative charge electrode, right? So why is this? This is because we learned that DNA or RNA molecule are negatively charged. Therefore, they will move toward the positive electrode. So if you put the gel in the wrong direction, then your samples will run out from the other side. So that's not going to work, okay? And then if you cut your plasmid DNA with restriction enzymes, after running the gel, you might see a picture similar to this, okay? Shown in the middle over here, this is called a DNA ladder or markers. So basically they are the DNA fragments with already known sizes, okay? They will give you a reference of how big the size of your DNA fragments. Okay, so let's see what information we can obtain from a DNA gel electrophoresis by answering several questions. So now I have a 5 kilobase pair of a plasmid DNA vector. Remember, we learned that plasmid DNA is a circled form, right? The DNA sample is very pure. That means there's nothing else. Based on the vector map, it has 2 EcoR1 restriction sites. How many DNA bands do I expect to see on agarose gel after EcoR1 digestion? So EcoR1, we learned it's a restriction enzyme. So it working as a scissors to cut the DNA, plasmid DNA. So you can stop here and think about it. And then we will, after you can pause and then we'll continue with the answer. So you actually can see probably 2 bands because as we mentioned, this is a 5 kb of plasmid DNA vector. You have here 2 EcoR1 restriction sites. So if you cut it on this circle, and then you will expect to see 1 fragment and another fragment, right? Okay, so let's put it this way. So if I only see 1 band on 2.5 kb, what does it mean? So you can pause here, and here is the answer. So say if the 2 sides equals one side is here, another side is here, it cuts the plasmid DNA into 2.5 kb. Then when you run it, these 2 fragments, they were overlapping with each other. They run at the same size. Therefore, on the agarose gel, you only see 1 band, okay? So both of them are 2.5 kb. Let's give you explanation. All right, let's see another possibility. So if I only see 1 band on 5 kb, what does it mean? Presumably, in this situation, we have the efficient cutting of EcoR1 enzyme. So what does it mean? Okay, here is the answer. So it's likely that if the 2 EcoR1 sites are very close to each other, then once you cut it, they will release very small fragments. When you run the gel, you don't see this very small fragment. It will run away very quickly, okay, very small. Then what you will see is a fragment that is almost equal on the 5 kb. Okay, so that's what happened. Of course, it can be caused by, say, the buffer condition is not correct, and your enzyme does not cut at all, or you barely incubate for enough time. Those are the all possibilities, but in this case, we don't talk about the like non-efficient cutting, okay? Then how about this situation? If I see 3 bands, 5 kb, 3 kb, and 2 kb, how could it happen? This is a little bit tricky. So this is what happened. In this case, it will be cutting inefficient. You have to be in this situation, because say if you see this 3 kb and 2 kb, that means add up together there's going to be 5 kb, indicating that the 2 EcoR1 site will generate 2 bands. One is 2 kb, one is 3 kb. So cutting here or here. And then why you still have 5 kb left there? If it's cutting completely, this 5 kb band won't be there anymore. You only see 3 kb and 2 kb fragments. Now, this tells you that this 5 kb is coming from incomplete cutting, okay? So that's how it happened. Okay, now we have learned the plasmid DNA and gene cloning, and I would like to introduce how we use these tools to build up our cloned libraries. Similar to a regular library that holds a lot of books, the cloned libraries also contain many DNA sequences from different sources. Okay, in a particular library, various DNA sequences are cloned into a common vector. Depending on the different sources of DNA sequences, there are genomic library, chromosomal library, expressed sequencing library, and many others that are not even included in this slide. Okay, for example, the genomic library is a collection of clones representing all of the DNA sequence of an individual. Right, and then the chromosomal library is a collection of clones representing all of the genes from one chromosome. And then expressed sequence library is a collection of clones representing the expressed genes from a given cell type. In this case, mRNA is isolated, and then cDNA is made by reverse transcription, okay? Because you cannot clone mRNA into a DNA plasmid vector. So you have to reverse transcribe it first and then clone the cDNA. These cDNA fragments are cloned into plasmid vectors, and all these clones from the library, okay? Cloned libraries are useful for many research purposes. Genomic sequencing, genetic screening are the major applications. Now let's see how a genomic library is constructed. This slide shows the steps of how a genomic library is constructed. Okay, you need the cells to get it started. There's different cells. The source of the cells can be like in vitro cultured cell lines or primary cells isolated from patient samples or animal models. Okay, so genomic DNA first is extracted from these cells and digested by restriction enzyme into these different pieces. Okay, the purpose of this step is to cut the genomic DNA into appropriate fragments so that they could be cloned into plasmid vectors. So they inserted these fragments into the common vector. Everybody in this library using the same vector. Okay. After cloning, the plasmid containing different genomic DNA fragments are transformed into bacterial cells. You can store these bacterial cells for later use, and you can also purify the plasmid DNA and store the library in a form of a DNA. So you see, these are the bacterial, their own genomic DNA, and these are the plasmid DNA you cloned in, okay? So this DNA can be used for downstream applications such as library sequencing. If you want to sequence of them, that's okay too. Before I introduce the next generation sequencing in next lecture, I would like to discuss about the traditional sequencing, namely the Sanger sequencing, okay? Sanger sequencing is the basis for development of next generation sequencing. So this sequencing is done based on PCR method. We already learned that, right? So however, instead of only using this DNTP, it's a deoxynucleotides in the normal PCR reaction, here we use the di-deoxynucleotides, also called a DDNTP. Okay, so you see the difference. This is a DDNTP. And you see there's one oxygen is missing. So so we using DDNTP added to the PCR reaction for this Sanger sequencing. The DDNTP do not have the hydroxy group on the nucleus sugar, and after incorporation into a newly synthesized DNA strand, further synthesis were stopped, showing this colorful nucleotides, those are ddNTP here. So whenever you insert it there, they will stop. Okay, so in this case, once the resulting fragments are separated by size using gel electrophoresis, the gel is scanned with a laser and the data can be analyzed by the software. Okay, each peak in a printout shows the light fluorescented by a labeled nucleotide. That's the ddNTP when it's labeled. So you can see here, for instance, here, this the first one is a T, stop here, and then C, stop here, and then another C, A, T, and then the machine will read that. So then you know the sequence. But then before the fluorescence labeling was available, when I was a college student, we used isotope labeling. So this is a picture of a typical DNA sequencing gel. The gel is very long, okay, maybe like 2 feet long, and ran for a long time. After drying the gel, the signal can be exposed to a film, and you could manually read the sequencing result, okay? So it is hard to imagine that after a little more than 2 decades, there is such an improvement on the technologies. Currently, the sequencing cost for each DNA sample is about $3. I mean, it's about $3. Sometimes like $3.50. And usually you can get the sequencing result the next day. We have talked a lot about gene cloning. One question is, can we clone the whole animal or even human being? So let's first take a look at some definitions. As we discussed, clones means genetically identical molecules, cells, or organisms all derived from a single ancestor, okay? So clones are not only applied to DNA molecules, it can also refer to organisms, right? So cloning is the process of producing identical copies of these biological entities. So yes, scientists have already successfully cloned animals. There are many true cloning methods. One is called embryo splitting. Another is called a nuclear transfer. Okay, let's discuss them individually. This slide shows how embryo splitting is done. So this is easy to understand because it naturally happens. So identical twins are actually embryo splitting, right? So at early stage of embryogenesis, the cells of embryo, this is also called blastomeres, are still totipotent, which means that these cells can develop into a full animal even when they are separated, okay? Therefore, this one single blastomeres are removed from the embryo, which under suitable condition continue to develop like undivided embryos. So the embryos are implanted to surrogates, and these produce identical offsprings. So these are surrogates mom, and then their babies, although these moms look different, their babies are all the same, okay? Embryo splitting has been successfully applied in livestock breeding, but not in human medicine as a result of legal restrictions and more complex development processes in human cells. Nuclear transfer is a completely different procedure, okay? It is also called SCNT, that means somatic cell nuclear transfer. As it's named, this technology needs both an oocyte, means the unfertilized egg, and a somatic cell. The nucleus of the oocyte is removed. So that's actually called the enucleated egg, okay? So the nuclei is removed and it's replaced with the nucleus of somatic cells. Like for example, there'll be skin, heart, or nerve cell, okay? So these provide the nucleus, this provide the cytosol. And then these two entities fuse together to become one, and the factors in the oocyte will cause the somatic nucleus to reprogram to a pluripotent state. Okay, so the cells were fused by an electric impulse and divided to form an embryo. Okay, this resulting embryo then is implanted into a foster mother, resulting in the birth of an animal. So let's watch a video to show how this method is done and with more details, okay?
Hey there! Welcome to Life Noggin. If you didn't already know, Ronbus is my best friend. He's great, he's really good at soccer for some reason even though he's never played, and I want more friends just like him. Exactly like him. It's easy to clone in my world. My animator can just copy and paste and voila, I've got 20 best friends. I am gonna need way more guacamole now. But what if you wanted to clone your best friend? Or even yourself? How would that even work? Cloning is the process of producing a genetically identical copy of a living organism. It can be done on many scales, from replicating basic DNA to a complete genetic copy of a living thing. Scientists have cloned a bunch of animals in the last 100 years, most notably the infamous Dolly the sheep in 1996. Now we've got cloned cows, horses, cats, and dogs running around, and just recently, 2 cloned monkeys joined the party. This party smells really bad, guys. If we could just clean some stuff up a bit. If we can clone primates just like monkeys, can we do it with humans? The most successful and well-known method of cloning full living things is somatic cell nuclear transfer. Scientists take an ovum from the female of the species they're cloning and remove all the DNA from inside. Then they take the nucleus of a somatic cell, like a skin cell, from the thing they want to clone and insert it into the egg. The egg is then implanted into a surrogate who carries the egg until it's born. Now that scientists have cloned a non-human primate, we're closer than we've ever been to cloning a person, but there's still a ways to go. The success rate for these monkeys was super low, as it often is with cloning. The scientists started out with 127 eggs and only ended up with 2 monkeys, meaning their success rate was only 1.6%. One of the biggest struggles with cloning primates has to do with spindle proteins. They're crucial in cell division and development, and in humans, they reside in the nucleus of an egg. So when the nucleus is removed to make room for the new DNA, those spindle proteins are removed too, making embryonic development that much harder. Say we found solutions to biological issues like this, navigated our way through the complex ethical implications, and successfully cloned a human. Would your clone be just like you? If you're 5'10" with brown hair and blue eyes, maybe your name is Steve, who knows, you'd expect your clone to look the same, right? Well, you may be surprised to learn that your clone would look completely different. Your appearance is based on genes, yes, but it also depends on the fetal environment you grow in, and which genes are turned off and on. Your clone would also be very unhealthy. Many cloned animals suffer from defected brains, hearts, and livers, and immune system problems. In lots of clones, the tips of their chromosomes shrink quicker, meaning cellular aging happens faster than normal too. So your clone could be really sick and die prematurely. Plus, your clone could have a completely different personality than you do. While your personality is based on some genetics, it's also formed through experiences and upbringing. Just because Ronbus is super friendly, motivated, and likes puzzles, doesn't mean his clone would. A Ronbus clone could be mean, dishonest, and hate puppies. So unless your clone had the exact same development and experiences you did, it really wouldn't be you. With all this being said, would you want to clone yourself still?
So this slide shows Dolly the sheep together with her surrogate mother. Dolly was cloned by fusion of a adult cell and a enucleated egg. Dolly was the first mammal cloned by nuclear transfer. Dolly lived 6.5 years and was euthanized because she had a progressive lung cancer and severe arthritis. The average lifespan of Dolly's species is about 11 to 12 years. 11 to 12 years. So it is not known if the disease had a connection with Dolly being a clone, although Dolly's telomeres were actually short. In this lecture, we have discussed the many biological technologies. Now I would like to do a virtual experiment, okay? So we learned about androgen receptor AR, which is a transcription factor that is very important for male sex development. Say, if you need a recombinant AR protein for your research, now you have a prostate cancer cell line named LNCAP, which express AR. In this case, we say this cell line is AR positive, okay? Can you think about a way to produce AR protein using this LNCAP cell line? And also, using the knowledge we learned from today's lecture. Please think about it, and I will show the answer in our next lecture, okay? That's all for today.